Part 3 of color perception. We're going to address a number of different theories of color vision. We're going to start with these basic theories of how color vision works. So the first one's going to be trichromatic theory of color vision. Tri meaning three, chroma meaning three colors, chroma meaning colors. That Young and Helmholtz, we will talk about their theory for a little while and provide some evidence and some examples, and then opponent process theory of color vision discusses those four colors that we mentioned before, so red and green and blue and yellow, and talks about them as having these opponent processes, and we're going to discuss both of those. And then we'll talk a little bit about color constancy and lightness constancy, and with color constancy we will address or talk really briefly about Retinex theory, which I don't think it's named as its theory in the textbook, so I might not end up calling it Retinex theory, but that is what it's known as. Is that the idea is that the cortex, that X comes from cortex, is making comparisons across the retina, which is why we see those contexts as being so important, which we've seen before. We're going to start with trichromatic theory of color vision. So tri meaning three, chroma meaning colors, that three basic colors or color mechanisms can explain the range of colors that we see. This was really originally proposed by Thomas Young and then formalized by Hermann von Helmholtz. He gets a lot of the credit for a lot of things, partly because he formalized information and wrote it down and systematically examined questions, but he also has he had the prestige, right? So he was one of those polymaths, physicians, physicists. He understood a lot of things about many different fields, and so I think it is that we still use the vision charts and things. A lot of what we still use came from Hermann von Helmholtz. And we saw him in object recognition when we talked about his theory of unconscious inference. So he's actually shown up a few times in the textbook. But they basically stated that we have these three basic color mechanisms, and those three colors are going to be able to explain color vision. And they discovered this through metamers, things that are physically in the environment. The physics is different, but perceptually they are identical. So behavioral support for trichromatic theory of color vision has been around for over 100 years. And these color matching experiments, they were designed and they were studied by John Clerk Maxwell, who gets a lot more of his attention for his understanding of electromagnetism and theories sort of just about the physics. But he did these color matching experiments where you would see a monochromatic light in the test field, and so let's just say it's 500 nanometers, but you would systematically see 500, 510, 520, like that. And so we're just showing 500 nanometers as an example. And so if they gave a participant one of those three lights, let's even just say 560 nanometers, and they can make that light darker and lighter, right? They have the ability to say how much saturation of this light do I want. 560 nanometers, there's nothing you can do to create that 500 nanometers. It's just not the same color, our experience. We add another one, so let's say 420 nanometers, and now with those two together, I can match some of those test field single monochromatic lights, but I can't match them all. But they gave them three. So 640 nanometer light, a 560 nanometer light, and a 420 nanometer light. With three of those, then I can match any monochromatic light in the test field. And so that was the behavioral support for, it looks like we have three basic color mechanisms. And again, you can see why these are metamers, that one of them is a monochromatic light, and the other one is I'm matching that using three separate lights. it wasn't until the 1960s that physiological evidence came along to support that behavioral evidence. So actually, these two theories of color vision, the people argued against each other for about 100 years. We had the behavioral evidence for both, and we didn't really know what the answer was. But here, researchers found photopigments that respond maximally to short wavelengths, so at 419 nanometers. They found other photopigments that respond maximally to 531 nanometers, which they called the medium wavelength cones. And they found another set of photopigments that respond maximally to 558 nanometers, which they called the long wavelength cones. They also, in the 1980s, found some genetic differences for coding for these proteins for these three different photopigments. So we have found the genetic evidence in addition to what's happening at those photopigments that they were measuring from, or measuring the absorption spectra of the cones, of the visual pigments in those receptors. Are three color mechanisms necessary for color vision? Yes, for our range of colors. For what most of us see, three color mechanisms are necessary. But no, we could see color with just two. And I'm going to go through this, so I'm going to do this kind of almost like a proof, if you will. And if you don't really like math or numbers, you might want to stick your fingers in your ears, but you don't need to know the numbers. I'm just using them so that I think it's really helpful. And they've changed the example in the book, and I like the way they talked about it better with this example, even though it seems maybe they changed it because it seemed a little sexist, and I'm sorry about that. I don't love the example, but I like the way they talk about and explain it better than the new way of explaining it. So we're going to look at J, who has one photopigment, and that photopigment absorbs 10% at 550 nanometers, and it absorbs 5% at 590 nanometers. As we look at those kind of almost normal-ish looking curves that we saw for those absorption spectra of the cones. And so if we look at Mary and Barbara, who were shopping at a monochromatic dress store, and Mary is wearing a 550 nanometer dress, and she is standing in light that is 1,000 lumens, which these are arbitrary numbers, don't take them to mean anything. But so what's happening with that cone is it's absorbing 10% of that, is it's going to fire at a rate of 100. And we can ignore hyperpolarization or making this more complicated, but I'm just going to say it's going to fire at a rate of 100. Okay, look at Barbara. She's in 1,000 lumens of light. She has on a monochromatic dress. That is 590 nanometers. And because this one cone is absorbing at 5% of the 590 nanometers, that cone is now firing at a rate of 50. But what happens when we put Barbara in twice as much light? Now she's in 2,000 lumens of light. That's being reflected into Jay's eyes, and he's getting 5% of that, and 5% of the 2,000 now is 100. And so Barbara, in twice as much light, that color that she's wearing, that wavelength that she's wearing, is going to look the same as Mary when she's standing in less light. And so that's why if we only have one cone or one photopigment, we're going to have just various shades of gray. But let's give Jay one more cone. Now Jay has the cone that he had before, and he also has this cone, which absorbs 5% of the 550 nanometer light and 1% of the 590 nanometer light. So we can think of this as kind of like the medium and long wavelength cones. Now, Mary's at 1,000 lumens of light. That first photopigment's getting 10% of that, so it's firing at a rate of 100. The second photopigment here is getting 5%, so it's firing at a rate of 50. Barbara is in 1,000 lumens of light in her 590 nanometer dress, and so that cone is absorbing 5% of that and firing at a rate of 50, and the second cone is absorbing 1%, so it's firing at a rate of 10, and it looks like Barbara looks different than Mary. But we saw what really made Barbara and Mary not distinguish between shades of gray was if we double the light, it looks the same. Well, what happens when we double the light? Now Barbara's standing in 2,000 lumens of light. That first cone, like it did before, is getting 5%, and so it's firing at a rate of 100. The second cone is getting 1%, and so it's firing at a rate of 20. So in 1,000 lumens light, the 550 nanometers, I have this ratio of 50 to 100, whereas Barbara in 2,000 lumens of light, 590 nanometers, I get this ratio, 20 to 100. So now it's the ratio, right? That 50 to 100 is different than 20 to 100. And so now we are distinguishing between those two wavelengths, or those two experiences of color. And so we can talk about what happens when we have a defective or absent photopigment. And this, I'm going to say, this is not really evidence for trichromatic theory of color vision. This is something that, as we learned about trichromatic theory of color vision, it informed our understanding of these color deficits. So we knew they existed, but we didn't exactly know why. So we have a number of different types of color deficiency, and we can determine or figure out whether or not somebody has a color deficiency by using these Ishihara plates. And you'll notice that I usually ask students their experience, but here we are. It's just me in a room by myself with a microphone. So if I ask you what you see in A, mostly students say 74. I will say I have had students with color deficiency at least in 5 or 6 classes now. And so I ask my students, all of them, what do you see in B? And I get this 21, this kind of question. It's hard to tell. Those light green don't really distinguish as well from the dark green, kind of olivey color, as we saw with the red and green. And my students with color deficiency tell me A and B look the same to them. And what we are seeing in B is 74. Only it is showing us what a person with color deficiency would see if they were looking at A. They're not distinguishing between reds and greens. Whether or not this is actually what they see, I'll talk about it a little bit more. But another way to see if somebody has a color deficiency is to use a color matching study. So if you can match any wavelength in the test field with any one wavelength in the comparison field, then you're missing at least 2 photopigments. If you have 2 photopigments and you're just missing 1 photopigment, then you need 2, right? You need 2 of those different wavelengths, and you can adjust those to match any wavelength, single wavelength in the test field. And I, as a trichromatic vision, like person with regular color vision, I might be behind you saying, yeah, those don't match, but you see them as matching. So they only need 2. So we can also get that from color matching. I'm going to give some examples and go through some of this a little bit more quickly. So our inherited color deficits are defective or absent photopigments. Dichromats, this is for most of our dichromats, this is on the X chromosome, so we see more males with this particular color deficiency than females, and they're missing a single photopigment. But when I say most, I'm going to show some examples. We also do have monochromats, which is extremely rare. 10 out of 1 million people, they're missing 2 photopigments or missing all of their cones. They do have worse visual acuity, and they're sensitive to bright lights. Their vision is basically based on the rods, which we've talked about as being helpful in dim light, but they're needing to regenerate when we're in light and brighter lights. So we're not really using them, and they're not giving us detailed vision because of that convergence. So some examples: missing the long wavelength cones, protanopia. You don't need to know these names, but here they are in case you want to. 1% of male and 0.02% of females. And as I move to the next one, people missing medium wavelength cones, you can see that those two cones together are letting us to see these distinctions, that the picture on the right, where we're kind of really missing the reds and yellows and that distinction from green, it's the picture on the right is going to look very similar for, get it to go, deuteranopes. So people with deuteranopia missing the medium wavelength cones, and it looks kind of similar because, again, I have one cone up there distinguishing those medium and long wavelengths. This is in 1% of males and 0.01% of females. And finally, trichromatic, where we think they're missing the short wavelength cones. It kind of makes sense. They're not, and they don't have that more blue-green distinction. And this is just much more rare. It's not on the X chromosome. It's different. And then monochromats, where they are truly in a grayscale world, a black and white kind of world. And they are missing either two photopigments, so they only have one cone helping them in the daytime, and so we're just seeing shades of gray, or they're missing all of their cones and they're truly just using the rod system. Again, really quite rare. We can compare ourselves to our animal friends, where many mammals are dichromats. Even some monkeys are trichromats. Goldfish are trichromats. Honeybees and birds are tetrachromats. And if you go look that up on Google and get some pictures. It is so cool that flowers that are just yellow to us basically have a bullseye showing them this is where the pollen is, so they see their food very clearly. And I did go rushing through a little bit of that extra information because I want to get to this other theory of color vision in this video. So trichromatic theory of color vision, it explains a lot, but there are still questions and phenomena that trichromatic theory of color vision cannot answer. And so again, this argument went on for about a century. Ewald Hering in the late 1800s, early 1900s, he suggested that we want to talk about 4 primary colors, red and green and blue and yellow, and that we need to talk about how they work in these antagonistic kinds of pairs, where red and green seem to have opposing responses and blue and yellow seem to have opposing responses. And he took a lot of really phenomenological evidence in looking at the color wheel, where red can be, we can think of red as being across from green and having no green in it, and we're not really putting green in it. If we start to add blue, we start to add blue, we go to purple and get towards blue. If we start to add yellow, we go to orange and go to yellow, and then we have completely absence of red, we get to yellow, and then we can start adding green. People do complain a lot about the color wheel, partly because it's phenomenological and partly because you could change those colors out with cyan and other colors. So other behavioral evidence are these after image demonstrations, which I'm going to go ahead with the Hurwitz and Jameson, and you can just, if you're looking at the fixation cross and you give yourself a little space there, I can talk for almost 30 seconds or more, but you can pause it, and you want to keep your eyes very still. You can have a little astigmus, your eyes tend to move, but as long as you're not looking off, looking at a watch or your phone or text, as long as your eyes are basically forward, this will work. Again, I'll suggest to pause it for a moment. So you can do this for a good 30 seconds or so. I'm going to move on. So if you did that for a long enough time and you kept your eyes there, you should be seeing, and I didn't do it for very long, and I have a very, very faint, what looks like a red, white, and blue flag. So what happened? I usually ask students, what happened? In one semester, I had this kid from biology rolled his eyes and he said, "We fatigued some neurons." And everybody in the class was like, what? But that is what we did. We fatigued the green, and so we ended up with this red after image. We fatigued the black, so we ended up with this white after image, and we fatigued the yellow, so we ended up with this blue after image, and so we got the red, white, and blue flag from the green, black, and yellow. That's hard to say. You can do that. And with these, if you want, you can go back and forth with that if it's fun for you. So the proposal was we have 3 opponent mechanisms, a black-white, a red-green, and a blue-yellow. And you can see the excitatory versus inhibitory. Again, this was the behavioral evidence, and this was the proposal. But eventually, from the behavioral evidence and that argument, there was eventually a time when they received the physiological evidence or the physiological support, where they were recording from retinal ganglion cells, that's what the RG cells, and lateral geniculate nucleus cells, so the LGN cells. And they found these neurons that had these opponent mechanisms, where if I look at the B plus yellow minus, so blue excitatory, yellow inhibitory, we see the top, not seeing anything. We have a spontaneous firing. I present a 450 nanometer light. We see this excitatory response, so lots of impulses. 510 nanometer light at green, maybe a little bit of response, but not much. 580 nanometers up at yellow, it looks like inhibition. 660 nanometers up at red, it looks like inhibition. And this has been visualized in a different textbook, but I liked it a lot. If you notice what's going on there with this particular bipolar cell, that it's receiving inhibitory information through that horizontal cell. The long and medium wavelength cones are having excitatory synapses with the horizontal cell that's going to give an inhibitory information, as is the short wavelength cone, but the short wavelength cone is also directly giving excitation to the bipolar cell. So this would be how we have that blue versus yellow antagonism. And this was discovered in about the 60s. So Goros in 1968 suggested or seemed to find that we had red center excitatory, green inhibitory surround, red inhibitory center, green excitatory surround, green excitatory center, red inhibitory surround, and green inhibitory center, red excitatory surround. So we had all those center surround receptive field possibilities, all of them. And he suggested that we had a blue excitatory center and a yellow inhibitory surround, but now they've discovered all 4 of those possibilities, which only makes sense for the blue-yellow antagonism as well. So ending this nicely with my 2 theories of color vision, of really how color vision works. If we ask which theory is correct, I'm hoping it's really clear that both theories are correct. That's why the behavioral evidence worked for each of them throughout those years. But now that we have the physiological evidence as well, we can say that, look, it looks like trichromatic theory or trichromacy occurs because we have these 3 kinds of cones, these 3 kinds of photoreceptors with their own visual pigments that respond differently to the different wavelength information. That's why it looks like we have these 3 color mechanisms. But we also have opponent processes, and that's occurring at the later levels because of that integration of inhibitory and excitatory signals that we talked about is happening in those between those neurons in the eyes, going from the photoreceptors to the retinal ganglion cells, and then really being maintained in the lateral geniculate nucleus cells.